Student, welcome to my channel. Today we will be learning about megasporogenesis, the process by which the megaspore develops inside the female gametophyte or the embryo sac is known as megasporogenesis. Now, a young ovule has all diploid cells in this, and the tissue present in the young ovule is known as the nucellus tissue. Now, any of the cell inside the nucellus tissue becomes dominant and begins to function as a megaspore mother cell. I repeat here, megaspore mother cell is a diploid cell. Now, the megaspore mother cell, now it will undergo meiosis or the reduction division. As a result, what happens? It will give rise to 4 haploid cells, and these haploid cells will be arranged in a linear manner in this way. So I repeat, this is one of the cell of the nucellus will function as the MMC, which by meiosis division will give rise to 4 haploid, these are all haploid megaspores, and they will be arranged in a linear manner. Now, out of this, any three will degenerate, leaving only one functional megaspore. Only one megaspore will remain functional here. And now, the rest of the development will be carried out only by this functional megaspore. Now, the next steps comes like this. Now, if I draw the megaspore in this way, so this is the megaspore and this is a haploid nuclei present in it, and this megaspore will now undergo division in this way. Only the nucleus of the megaspore will undergo repeated free nuclear division. So this is the first division and this is the second division. We get 4 nuclei, and this is the third division where we are getting 8 nuclei. Here the point to be noted is that the nucleus is only undergoing simple mitosis division here. Only mitosis division, because this is always already an haploid nuclei which is undergoing repeated mitosis division, and after the third division we are getting 8 nuclei inside the megaspore. Now what will happen? This nuclei will now be differentiated in this way. 3 will be remaining at one end and 3 on the other end, and 2 nuclei will be arranging themselves just in the center. Now the 3 nuclei on both the ends will be now surrounded by the cell 1. I mean, now we will be calling them as cells. The 3 cells which are at the micropylar end, near the micropyle, out of these 3 cells which are at the micropylar end, one will be functioning as the egg cell, that is an haploid egg cell, and rest of the 2, I mean this and this, these 2 cells we will be calling as synergids. These 2 will be calling as synergids, and the opposite pole, which is known as the chalaza, this pole which we will call as the chalaza, these 3 cells which are present at the chalaza or the chalazal end will be known as the antipodal cells. They are antipodal cells. They are the antipodal cells, right? And the 2 free nuclei which we see in the middle of the megaspore, this is the megaspore in the middle, we will be calling them as the polar nuclei. I repeat, the polar nuclei will not be surrounded by any cell wall and they will be free in the middle. Now, the stage at which we are finding within the wall of the megaspore, we get 7 cells. That means 1, 2, 3, then on this end 1, 2, 3, that means 6, plus this. Now these polar nuclei will be surrounded by a single wall when it matures. So what we are getting inside the megaspore, there are total 7 cells. Out of the 7 cells, the 6 cells are having one nucleus I mean, and the central जो cell है, it is having 2 nuclei. So in this stage of the megaspore development, what we find is that it is having 7-celled 8-nucleated condition. At this stage we will be calling this as the female gametophyte. Now this will be known as the female gametophyte. In other words, we can also call it the embryo cell, which we also call as the embryo cell. So now in this way we have learned how the female gametophyte or the embryo cell development takes place. The next video we will be learning about the fertilization process. Till then, keep learning. Thank you so much.